Introduction to the topic drug design. Drug design is science, technology, and art all in one. An invention is the result of a creative act. A discovery is the recognition of a pre-existing reality. Design involves both processes, but emphasizes a focused approach based on existing knowledge and technologies. In addition, the creativity and intuition of the researcher play a crucial role. Drugs are all substances that exert an effect on a system by inducing a particular effect. In the context of this book, they are substances that exhibit a biochemical or pharmacological effect, in most cases as drug medications, that achieve a therapeutic effect in humans. The idea of rational drug design is not new. Already more than a century ago, organic compounds were synthesized in a targeted manner in order to produce new drugs. Scientists have always worked with models and design hypotheses. In the early days, these models were often flawed or even wrong. As our understanding of the molecular basis of diseases has steadily improved, the models and hypotheses have become better and more reliable. However, we are still far from the dream of designing a finished drug using rational concepts on the drawing board. In the case of artistic design, such as a poster or a commodity, or in the case of engineering, such as the design of a car, a computer, or a machine, the result is usually directly predictable. In contrast, the design of active substances is even today not sufficiently predictable. The consequences of even the smallest structural changes of the biological properties and the mode of action of a biologically active molecule are too multifaceted and yet too poorly understood. Until now, scientists have relied on the principle of trial and error in order to find new medicines. The rules empirically derived have resulted in a knowledge base for the rational design of active substances, which individual researchers have put into practice with greater or lesser success. Today, many new methods from genetic engineering, structural biology, biophysics, and informatics are available for the search for novel drugs. The work of recent years has contributed above all to our understanding of the molecular mechanisms of action of many known drugs, although these compounds were often previously found by simpler methods and sometimes even discovered by chance. This book attempts to present many of these modes of action or mechanisms. Methodological advances in experimental structure determination now allow the routine determination of the three-dimensional structure of proteins, RNAs and DNAs, as well as their ligand complexes. As shown in many of the illustrations in this book, in combination with the commented videos provided via the internet and accessible via QR codes, they make a very important contribution to the reader's understanding of the targeted design of active agents. Three-dimensional, 3D, structures down to atomic resolution are known of more than 1 million small molecules and more than 200,000 proteins, nucleic acids, and protein ligand complexes. Their number continues to grow exponentially. Methods for predicting the spatial structures of small molecules are mature. Semi-empirical and quantum chemical calculations on active agents are routinely used. The sequencing of the human genome is complete, and new genome data from other organisms are being added weekly. Recently, significant progress has been made in predicting the 3D structures of proteins from their amino acid sequence. With the help of artificial intelligence, computer programs evaluate the enormous treasure of experimentally determined structural data. What is still lacking is a comparably powerful and detailed database that reveals the structure of water molecules in the interior and on the surface of biomolecular complexes. Yet, their properties are essential for understanding how drugs bind and act. Predictions of the protonation states of amino acids in protein binding sites and of ligand functional groups, particularly their changes during ligand binding, are still in their infancy. The potential of RNA molecules for drug therapy is slowly being recognized. Concepts for structural biological classification and de novo prediction of the spatial structures of RNA molecules, including their solvate structures and protonation states, still need to be developed. Only then will it be possible to use machine learning and artificial intelligence to provide solutions for drug design through computer algorithms, similar to the current protein structure prediction from sequence. The structure-based and computer-aided design of new drugs is now an integral part of practical drug research. Thanks to a dramatic increase in computing power, computer programs now support the search, modeling, and targeted design of new active agents. The mere increase in throughput can possibly enable these techniques to exhaustively design and filter for potential drug candidates. However, to take the methodology to a higher level of reliability and relevance, a deeper understanding of drug-target interactions is essential. How do the local water structure and the mutual polarization of the binding partners affect the biological properties? Restricting ourselves solely to the interactions of the pharmaceuticals with the residues in the binding pocket of a single biological target structure also does not adequately capture the complexity of drug action. The binding kinetics and residence time of a drug at its target structure are just as important as pharmacokinetic, toxicological, and metabolic properties. How important are modulations of the dynamics of the formed complexes for drug action and therapeutic success? What other target structures are influenced in addition to the actual target? How troublesome, or perhaps even how important, are adverse side effects? When can a patient be offered personalized therapy and individualized dosing? Today, the gene sequencing of each and every one of us is possible at a financially affordable cost and in a reasonable amount of time. But how can this knowledge be used for personalized medicine? Future drug research will have to answer these questions for patients without losing sight of the cost-benefit ratio. So why is the development of a new drug still so complicated, and why has the time it takes hardly been reduced in the last 40 years? The cost of developing and launching a drug to market has risen steadily and continues to do so. The current cost range is between $200 million and $4,000 million. Only large pharmaceutical companies, especially those in the top sales and profit segment, can still afford such an investment. There is always the risk of failure in the late stages of clinical trials or of misjudging of the therapeutic potential of a new compound. There are often enough setbacks. On a more optimistic note is the observation that drug discovery is now tackling much more challenging targets. 10 or 20 years ago, these were still considered inaccessible or simply undruggable. Examples include the successful disruption or targeted enhancement of protein and protein contact surfaces by small molecules, or the targeting of over-regulated or pathogenic proteins for selective proteolytic degradation. The body's own immune system is enhanced to selectively eliminate virus-infected or degenerated cancer cells. There is, however, a crucial hurdle to overcome when using drug molecules to interfere with the control mechanisms of biological processes. In order to compete with the natural ligands of enzymes and receptors, or to mimic their properties, they must be sufficiently precise and efficient in terms of both the mechanism of action and the site of action. In the case of endogenous active compounds, our organism uses different principles. Substances such as the body's own hormones act predominantly systemically, meaning they are released at one site in the body and transported via the bloodstream to one or more completely different sites of action. It is only there that they develop their effect. This requires a high degree of specificity and selectivity of action. Substances such as neurotransmitters are also used by our body in many places for very different tasks. This can only be achieved in a controlled manner if they are used strictly locally. Our organism uses a highly developed spatial compartmentalization for this purpose. Substances such as neurotransmitters are formed close to where they are needed, where they exert their task, and then they are immediately removed again. Drug research is also aimed at interfering with these mechanisms and possibly correcting them, but we want to use substances that can be taken orally if possible. As with hormones, this is only possible if a very high specificity and selectivity can be achieved. In addition to this, sufficient stability and bioavailability from the gastrointestinal tract must also be achieved. Under these conditions, it is extremely difficult to achieve a tailored therapeutic influence on target structures for which the body itself uses only relatively small and unselective ligands. Inevitably, our active agents must become larger in order to achieve the required selectivity of binding. It is obvious that efficacy profiles of such substances will be somewhat different, and usually they will increase in size for selectivity reasons. All these aspects open up enormous perspectives for drug development, but they do not make it any easier today. The purpose of this book is to describe the development of new drugs under these difficult and ever-changing conditions. Design methods are presented, and drug development is illustrated with known mechanisms of action and selected case studies. Drug discovery is a multidisciplinary field in which chemists, pharmacists, physicians, technologists, molecular biologists, biochemists, pharmacologists, toxicologists, and clinicians work together to pave the way for a compound to become a new therapeutic drug. For these reasons, the majority of drug development still takes place in industry. Only industry has the financial resources and, above all, the organizational structures needed for all the disciplines involved to successfully work together. Only in this way can research be channeled in a targeted manner. However, the basic principles and forward-looking innovations in drug research are increasingly being developed in academia, not least for reasons of cost and critical risk assessment. Interestingly, more and more university research initiatives are focusing on the development of drugs for infectious diseases, rare diseases, or diseases of the third world. The inevitably commercially oriented pharmaceutical industry in the industrialized countries has increasingly withdrawn from these areas. This is all the more alarming when one considers that our improved quality of life and increased life expectancy are largely due to the victory over devastating infectious diseases. The coronavirus pandemic that we have just lived through has brought this back to our minds in a very vivid way. Rising research and development costs, an already high standard of therapy in many indications, much greater safety awareness and, as a result, more stringent regulatory requirements have led to a steady decline in the number of new chemical entities, NCEs, introduced annually into therapy over the past few decades, in the 60s between 70 to 100 NCEs, in the 70s 60 to 70, to an average of about 50 in the 80s and 40 to 45 in the 1990s and the 2 decades of the new millennium. However, in addition to the expansion of indications for drugs that have been known for a long time, it is the new developments that have brought significant progress in therapy. A paradigm shift is often talked about in pharmaceutical research. This refers to the application of new technologies and knowledge. As far as the structure of the market is concerned, the process of concentration through company takeovers, acquisitions, and mergers into giant big pharma companies has slowed considerably. Fortunately, this has given way to a very dynamic, almost unmanageable scene of small, highly flexible biotech and startup companies that are stimulating pharmaceutical research with new, innovative strategies. Big pharmaceutical companies are outsourcing high-risk research concepts to these small biotechs and using their services up to and including the development of clinical candidates. In addition, prescribing practices are changing across the healthcare sector. In the past, the physician alone was responsible for therapy, sometimes in consultation with the pharmacist. Today, cost pressures, negative lists, health insurance companies, hospital or drugstore purchasing organizations, the ubiquitous internet, and even public opinion are increasingly influencing therapy. This book is a textbook on drug discovery, the principles of action, and the way new drugs are discovered and developed. It differs from the classic textbooks on medicinal and pharmaceutical chemistry both in its structure and in its aims. It covers the basic principles, methods, successes, and obstacles in the search for new drugs. Rather than discussing classes of drugs in terms of their indications, it focuses on the route to the active compound and the structural requirements for its action on a particular target protein or family of target proteins. As the title suggests, the book is aimed at students and scientists in the fields of chemistry, pharmacy, biochemistry, biology, and medicine who are interested in the art of designing new drugs using knowledge of the structural basis of their activity at the site of action. The first part begins with an introduction to the history of drug research. This is followed by a description of serendipity as a concept in drug discovery that is difficult to plan but always highly valued. Selected examples from classical drug research are presented as examples. A discussion of the fundamentals of drug action, the thermodynamics of ligand-receptor interactions, and the influence of the three-dimensional spatial structure of a drug on its efficacy round off this part. In the second part, the search for new lead structures and their optimization and the use of prodrug strategies are introduced. New screening technologies are discussed, as well as the systematic modification of structures using the concept of bioisosterism and a peptidomimetic approach. The third part describes experimental and theoretical methods used in drug discovery. Combinatorial chemistry has provided access to a large number of test compounds. Gene technology can produce the target proteins in their pure form and has helped to characterize the properties and function of these proteins from the molecular level to the cellular assembly to the organismal level. It has bridged the gap between understanding the effects of drug therapy on the complex microstructure of a cell and the systems biology of an organism. The spatial structure of proteins and protein-ligand complexes is accessible through X-ray crystallography, cryo-electron microscopy, and NMR spectroscopy. The structural principles of proteins and DNA are becoming better understood and are increasingly providing access to the binding geometry of drugs. Computational methods and molecular dynamics simulations, including complex conformational analysis, have also increased our understanding and modeling perspectives of targeted drug design. The fourth part introduces design techniques such as pharmacophore and receptor modeling, and discusses the methods and uses of quantitative structure-activity relationships, QSAR. Insights into drug transport and distribution in biological systems are provided, and various structure-based design techniques are presented. A drug design case study from the author's research concludes the first part of this textbook. The fifth part focuses on the core question of drug design: how do drugs actually work and how does this translate into the design of new drugs? Enzymes, receptors, channels, transporters, and surface proteins are divided into separate chapters and discussed as a group of target structures. The spatial architecture of the protein and the modes of action are used to explain in detail why a drug works and why it must have a particular geometry and structure in order to work. These chapters illustrate the contributions of structure-based design and medicinal chemistry optimization to the discovery of new drugs, and highlight different aspects of the drug discovery process. The concept of this book means that many important drugs are not covered, or are only mentioned in passing. The same applies to receptor theory, pharmacokinetics and metabolism, the basics of genetic engineering and statistical methods. The biochemical, molecular biological, and pharmacological fundamentals of the mode of action of drugs, which are important for understanding drug design, are only briefly discussed. Other disciplines, such as pharmaceutical formulation, toxicology, and clinical trials, which are critical to the development of a compound into a medicine and its use in patients, are not covered in this book. The selection of examples from therapeutic areas has been made subjectively and for didactic reasons based on case studies, and to highlight different aspects of drug discovery. An attempt has been made to provide a balanced presentation of drug design methods and their practical application. The interested reader need not read the book chronologically. If the reader is interested only in drugs and their mode of action, he or she may start with Chapter 22. The reader who is more interested in methodology, or the medicinal chemist who wants to learn the basics of drug design, can concentrate on Chapters 4 to 21. There are many cross-references throughout the text to help the reader find the passages needed for a more detailed understanding at a particular point in other sections. In the introduction, a bibliography list is provided of particularly recommended monographs and, in alphabetical order, journals and series on the subject, which are not mentioned individually in the later chapters. The reader will also find a summary of the most important aspects after each chapter. Suggestions for further reading and many original citations are given at the end of each chapter.